This podcast is going to focus on the integumentary system, and it's crudely going to be split into three parts. The first part, we're going to think about the epidermis, because really we're thinking about the skin when we think about the integumentary system. So we'll focus firstly on the epidermis, then we'll move on to the dermis, and then finally we'll talk about the hypodermis and the integument accessory structure. But of course, as you may well know, the skin is actually the largest organ in the body, and it acts as a mechanical barrier between the external environment and our underlying tissues. Its deep layers, where we've got the blood vessels, they could extend about 18 kilometers if they were stretched out. So there's a lot of blood vessels going around our skin. When we're talking about this, we're talking about the epidermis, dermis, and hypodermis. But technically, the skin only consists of the outer epidermis and the dermis. The hypodermis isn't technically part of the integumentary system, part of the skin, but we will be discussing it. And that's where that fatty layer is. So that is your hypodermis. So what do you already know about the functions of the skin? What can you think of? And I'd like you really just to press pause for a second and think for a minute or two. What do you know about the functions? So you know that it acts as a barrier and it acts as protection. But what else does it actually do? So press pause and have a little bit of a think. So of course it's involved in protection. So it's involved in cushioning our underlying tissues and organs. It's also very waterproof, which obviously helps us to prevent water loss, unless that is necessary for cooling. It protects us from chemicals, from heat, from cold, from microorganisms, and it helps to screen the harmful UV rays, in particular by the production of melanin by our melanocytes, which absorb that harmful UV. It's involved in the production of vitamin D, and it's involved in that first stage in the synthesis of vitamin D, where you get activation of or action of UV light on cholesterol. So that's the function of the skin. And to make active vitamin D, of course, you also require a functioning liver and a functioning kidney to fully activate vitamin D. And as hopefully you know, vitamin D, what's it involved in? Well, one of its primary roles is in the absorption of calcium and phosphate from your GI tract. So a vitamin D deficiency leads to the disease osteoporosis in adults or rickets in children. It's also involved in body heat and homeostasis. It regulates our body temperature through vasodilation or vasoconstriction of blood vessels. It's involved in sweat production and hence evaporation. And it's the evaporation that leads to cooling. And of course, it's got fat layers there to help with insulation. And it also excretes salts, water, and organic wastes. And finally, sensory. It's got multiple sensory receptors. It can respond to pressure, to pain, to vibrations, to heat, and to cold, as you will know. So let's have a little look at the structure of the integumentary system. So as you can see from the integumentary system, we've got the outer layer, which is the epidermis. And then we've got the inner layer, the dermis. And finally, we've got this hypodermis, which contains that subcutaneous tissue with all the fat that's present within it. We've got a lot of accessory structures. So those accessory structures include things like the hair, the sweat glands, the sebaceous glands that secrete sebum, for example. So before we move on to some of the functions and the structure of the epidermis, which again is shown in this image, you can see the epidermis there and you can see the dermis, and then moving down into the hypodermis. We'll just talk about some of those sensory receptors that are present. So we've got what are called free nerve endings. So you can see the neuron with its three little nerve endings. And these are involved in pain and temperature sensation. We've then got the Merkel discs, which you can see over on the right-hand side, nicely highlighted. And you can again see there within the epidermis layer, so they're very near the surface of the skin. So therefore, they're going to respond to a light touch because they're near the surface. So that light touch is stimulating your Merkel discs. So if you were to now, right at this second, stab a pin into yourself, it'd be stimulating these three nerve endings for pain or for temperature. If you were to just lightly touch your skin, you'll be stimulating the Merkel discs. You've also got the root hair plexuses, okay? And these basically, as you can see in this image, they entwine around the hair follicles. And because, of course, they're involved in the hairy parts of our skin, because not all parts of our hair has skin, just look at the palm of your hands. If you've got hair on there, I'd be worried a little bit. So again, you've got these hair receptor follicles, and there again, when they move, they stimulate these root hair plexuses, so therefore responding to light touch. They're helping in that light touch process or sensation. We've got our mucinous corpuscles. Again, if you're looking at these, they're fairly near the top, so they're responding to touch and to light pressure. But this is in hairless skin, so you don't find them in the hairy parts of your body. You've got the Raffinis corpuscles. They're, as you can see, a lot deeper down. So we're thinking about deep pressure sensation and also stretch. And then you've got your Pacinian corpuscle, which is also further down. So again, they're responding to deep pressure, to vibrations. And in terms of your visceral system, so your internal organs, they're looking at pain. They respond to pain, nausea, hunger, and fullness. So as we've already said, if we're looking at the structure of the integumentary system, we have the cutaneous membrane, which is our skin. It has this outer epidermis layer. And so this contains that superficial epithelium and then that inner dermis layer, which is made up largely of connective tissue. Now, on average, the epidermis replaces itself roughly every 2 months or so. And these outer layers of the epidermal cells, they're dead and they're flattened, while the inner layers of the epidermis are composed of cube-like shaped cells. These are living and they're rapidly dividing. But the epidermis hasn't got any direct blood supply, so its cells need to be nourished by diffusion of nutrients from the vascular system, which is present within the dermal layer that we can see here. So you can see how it spreads up throughout the dermal layer, but it doesn't go into the epidermis itself. So what is actually happening? Well, the newer forming cells that are forming at the lower levels of the epidermis are continuously pushing the older cells closer to the surface, further away from the nutrient supply. And because they're further away from the nutrient supply, they get less nutrients and they're more likely to die. But of course, they're getting that continuous wear and tear. And so they die and then start to become flattened, as opposed to those cube-shaped cells. The accessory structures that we've talked about, so things like your hair, for example, they originate in the dermis and they extend through the epidermis to the skin. So that's your hair, your nails, and your exocrine glands. Remember what the exocrine glands are? They're things like your sweat glands, producing sweat, and your sebaceous glands, producing your sebum. And as we've already said, we've got blood vessels that are present within the dermis itself. And the nervous system is responsible for the senses of pain, touch, temperature, vibration, etc., that we've already talked about a little bit. So remembering what we said, the blood is passing through the hypodermis and it passes into the dermis and not into the epidermis layer itself. And the hypodermis, as we already know, it contains loose connective tissue, it contains fat, and it contains those blood vessels that we've just been talking about. Just in case when you're reading around the subject a little bit, the hypodermis does have other names. It can be called obviously the hypodermis, but also the subcutaneous layer and also the superficial fascia. And it's located underneath or below the dermis. And as you might expect, the hypodermis is obviously the location for hypodermic injections. So the epidermis, this is something we've already touched on a little bit. The nutrients and the oxygen that they need, they have to diffuse from capillaries that are present within the dermis itself. And this is just taking a look at this the structure of the dermis and the epidermis. And we have two types of skin. We've got thin skin and we've got thick skin. And if you're looking at the thin skin, the epidermal layer is much smaller than in the thick skin, where it is much thicker. And again, with the epidermis, you can see it being peeled off in this part. You can see these epidermal ridges, and we'll talk about that a little bit later. So the epidermis, it consists of these avascular, because there's no vascular system, stratified squamous epithelial cells. And the most abundant cells are the keratinocytes, and these contain large amounts of keratin. And as I've already talked about, we've got thin skin and thick skin. The thin skin has 4 layers of keratinocytes, and the thick skin has 5 layers of keratinocytes. Now, the thin skin, that covers most of the body. And like I said, it's got 4 layers. The thick skin covers the palms of the hands, soles of the feet, where it is basically subject to the most pressure. And there are different layers that are there as well. So the stratum germinativum, very badly pronounced, I do apologize, which is the inner layer of the epidermis, that's got many epidermal ridges, which you can see there. And that's what makes up your fingerprints. So now we're moving on to a little bit of the structure of the epidermis itself and those layers that I so badly pronounced a second ago. So if we're looking at those 5 layers in this image, and we're looking at the thick skin at this point. So here's the surface, and you can see the first layer is the stratum corneum. I can say that one all right. You then got the stratum lucidum, said that badly, clearly. Stratum granulosum, spinosum, and then the one I definitely can't say, germinativum. There you go. And they're all, and this layer obviously is attached via the basal lamina, which again proceeds down into the dermal layer. So this germinative layer, this is the one that has got many stem cells. And those stem cells are able to, obviously, that's where we get that high level of replication going on. And this is attached to the basal lamina by processes or structures called hemidesmosomes, which basically form a strong bond between the epidermis and the dermis. As I said, this is the area where you get your fingerprints from. So the stratum germinativum, God, I'm bad at saying that, has got those epidermal ridges which make up those fingerprints, as I've already mentioned. We also have Merkel discs present in hairless skin, which respond to touch in this region, and the melanocytes that contain the pigment melanin are also scattered throughout this layer. We then move on to the next layer. So we're moving up the skin, so we're now onto the stratum spinosum. This contains roughly 8 to 10 layers of these keratinocytes, and the cells continue to divide, which helps to increase that epithelium thickness. The layer contains our Langerhans cells, which are active in the immune response. And really, these cells are tissue-resident macrophages. They reside in the epidermis, but also in the epithelia of the respiratory, digestive, and urogenital tracts. And they specialize in antigen presentation, and they're part of the skin immune system. The stratum granulosum, that's the next layer up, is the grainy layer. So if we're looking at it, you can see the little grainy layer that's present there. It doesn't divide, and it produces keratin. So the cells of the stratum granulosum, they produce the protein fibers, a variety of protein fibers, before dehydrating and dying. And remembering what keratin is, it's a tough fibrous protein, and it helps actually to make up other structures such as hair and nails. We then have the stratum lucidum. This is a clear layer, and it's only found in thick skin. So you don't see this layer in the thin skin, and it covers over that stratum granulosum. And the stratum lucidum cells, the layer is basically flat, dense, and filled with keratin. We then move to the stratum corneum, which is obviously the outer layer, the layer that's exposed on the surface of the skin. It's also called the horn layer. And the stratum corneum has about somewhere between 15 and 30 cell layers of keratinized cells, and these are shed and replaced every couple of weeks. So the stratum corneum, as is going to be quite obvious for you, being the outer layer, is obviously the water-resistant layer. And keratinization occurs on all exposed surfaces except for the eyes. So if we're looking at the skin life cycle, it takes somewhere between 15 and 30 days for a cell to basically move from the stratum germinosum to the stratum corneum. So now we're going to think about perspiration, or sweating as you probably know it. And we have insensible perspiration, and that's the loss of interstitial fluid by evaporation through the stratum corneum, the outer layer. Sensible perspiration is water excreted by sweat glands. So we have insensible and we have sensible perspiration. It's important that you understand the subtle differences between the two. So a question for you is really, will damage to the stratum corneum through significant burns or blisters, for example, lead to sensible or insensible fluid loss? So hopefully you thought about this. You press pause because obviously you have to think about it, and you came up that it's insensible perspiration that is taking place. So of course, water loss through the skin can obviously lead to dehydration, can be caused by lots of things. So damage to the stratum corneum, so your burns and blisters, as I asked you in that example. But also through immersion in hypertonic solutions such as seawater. So that's a very high concentration, okay, such that water is leaving the skin cells and moving into the seawater, basically trying to make the concentration of salts the same on both sides of the cell layer, if you like. So water gain, so hydration through the skin, may result from immersion in a hypotonic solution, such as your bath water. So that overhydration, of course, can lead to wrinkling and stretching of the skin. So that's why you come out all wrinkly when you come out of the bath. So it's all water movement through the process of osmosis. So what about skin coloring? When skin color depends on a variety of pigments, so such as keratin and melanin. So melanin is the brown pigment produced by melanocytes, but also blood circulation. So when you vasoconstrict and vasodilate, you have different amounts of blood going to the skin, as well as a variety of disease processes. So for example, when you're jaundiced, which is a buildup of bilirubin in the skin, your skin goes a little bit yellow. So keratin itself is an orange-yellow pigment, which is found in orange vegetables, and some people have taken too much keratin through a variety of juices, for example, and they get a slightly orangey tinge to their faces and their skin. It doesn't have any adverse effects particularly, but it has been known to happen to some people, because it accumulates in the epidermal cells and the fatty tissues of the dermis, which you can then see, of course, and it can be converted to vitamin A. But what about our capillaries and when we vasoconstrict and vasodilate to our skin? So I want you to just have a little bit of a think about that, have a read of this bit, and tell me what happens to the colours. So oxygenated red blood contributes to skin colour, as when blood vessels dilate, the skin reddens, and when skin blood flow to the skin decreases, skin pales. So it's nice and simple. You also get a cyanosis, of course, which is when you start to get a bluish skin tint, and that's caused by a severe reduction in blood flow or a severe reduction in oxygenation, and things like jaundice that we've already talked about. And so when we're thinking about skin coloring, we're really thinking about melanin and melanocytes. So melanin really is this brownish pigment which is produced by the melanocytes within the stratum germinativum. And it's stored in transport vesicles, these are called melanosomes, and transferred to the keratinocytes. As you know, melanin helps to protect the skin from sun damage, and skin color itself depends on melanin production, not the number of melanocytes, because it's thought that we all have very similar numbers of melanocytes. It's just that the amount of melanin produced by them varies quite dramatically. Like I said, there are other things that can cause changes in skin color. We thought jaundice and liver disease. You can be thinking about vitaglio, which is a lack of melanocytes and therefore a subsequent loss of color. You can be thinking about Addison's disease, and that's where you have autoimmune destruction of the adrenal cortex. It means you have a lack of cortisol, which is your long-term stress hormone. But through mechanisms that we'll talk about at a later stage when we think about the endocrine system, you basically get an increase in a hormone that has activity that will stimulate melanocytes and therefore leading to an increase in melanin production and darkening of the skin. So again, we can think about that at a later point. So really, if we're thinking about a summary of the epidermis, we're thinking, well, it's multi-layered. It can be 4 or 5 layers. It's flexible and it's self-repairing. Helps prevent fluid loss. It protects from UV radiation. It produces vitamin D, and it helps to resist abrasion, chemicals, and pathogens. Or can you actually remember? So here's a little question, a series of questions for you, and I want you to press pause for each one, and then we'll go through them in turn. People with albinism have a defect in tyrosinase, which is an oxidase that helps to control skin pigment production. In what epidermal layer is tyrosinase active? So hopefully you've said layer C, the stratum germinativum. I think I'm saying it right. I have absolutely no idea. But of course, that's where we have a pigment production. Next question. Again, press pause and then I'll read through it. A second-degree burn can result in loss of sensation, but doesn't necessarily affect motor control of the erector pili muscles, so they're involved in making your hair stand on end. Which of the following are damaged in a second-degree burn? Quite a challenging one for you, perhaps. So Pacinian, Meissner's, hair follicles, or Ruffini endings. So the answer is Meissner's corpuscles. So remember, these respond to light touch, and are found at the border of the epidermis and dermis, so they'll be damaged in a second-degree burn. Pacinian are deep pressure, hair follicles are deep, and ruffini are all deep, hence why the answer is mycinosis. So we've got something for you to look at and have a little bit of a think about as well. So scientists are measuring mechanoreceptor reactions to a variety of stimuli. What stimuli would you expect to generate the following response? So if we're looking at the response, there's very little response in the mycinoid and the Pacinian and the ruffini endings, but the Merkel discs are responding and the hair follicle receptors are responding. So are we thinking about constantly changing deep touch on a hairy skin? Are we thinking about sustained deep touch on a hairy skin, sustained light touch on a hairy skin, or constantly changing light touch on a hairy skin? So you've got to be thinking about this. So the answer is D. So this is light touch on hairy skin. So you're getting the Merkel disc and the hair follicle receptors. So you know it's going to be on hairy skin, which you didn't really have much of a choice about because that's the only option you had. And the Merkel and the hair movement sensations, something you might not know, they both adapt to sensation. Therefore, you need to change that touch required. You can't just keep your finger on there because it adapts and you no longer feel it. They no longer correspond. The deep skin receptors aren't being activated, which rules them out. As we know, the mycinoid responds to light touch, but only present on hairless skin. So it explains why there's no signal in this particular one.